So now, before we start this, what I want to tell you is that vitamins can be very, very, very complicated. Vitamins are cofactors. They're cofactors for enzymes. So enzymes will have their binding pocket, their active site, and they cannot work, some enzymes, unless they have a cofactor. That cofactor fits into the binding pocket, and it's going to mediate the entire reaction sequence that that enzyme performs. That means that all vitamins have a rather complex chemistry that they perform. So there's two ways to look at vitamins. One is we can go through the detailed, gory mechanism. Thank you, that's kind of what I'm thinking. No, don't go there, don't go there. Or we can talk about the medical related aspects of vitamins, and then I can give you a little sprinkling of mechanisms just so you know, to see how these things function and anchor that information based on the enzyme and the clinical presentation. So that's kind of what I elected to do. Now, that being the case, what kind of questions would I ask you on an exam? Let's see, there's several on your summative involving vitamins, and what they're looking at primarily is that a deficiency, this person presents with this. For example, the first one we're going to talk about, vitamin B1. This is thiamine. The classic presentation is beriberi. That's a step one piece of information. That is not a step one piece of information for me. But that's something you got to know for the step one, that beriberi is a thiamine deficiency. What I want you to know is, where does this vitamin affect? What enzyme does it affect, and what are the consequences of that? So here, this is a perfect one, B1. This is, person presents with muscle weakness, fatigue, tiredness. I mean, it sounds like a medical student, right? And you guys, I'm sure, are filled with vitamin B1. But those kind of symptoms, I'll try to lead you down a way saying that there's a vitamin deficiency, or say there's a vitamin B1 deficiency you identify in this person. Which of the following enzymes might be compromised, leading to these clinical phenotype? Okay? So it's recognition of the vitamin with the clinical presentation, with the enzyme it affects, and the effect on that enzyme, how does it come full circle to the clinical presentation? That's kind of the world of vitamins that I've constructed. So that being said, vitamin B1 is thiamine. Weakness, pain, weight loss, muscle fatigue, muscle cramping, tiredness, lack of energy. These two enzymes, pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase, those are the two enzymes you need to know connected with vitamin B1. And that's because both enzymes perform this same chemistry. So let's say this is PDH. We looked at PDH. And remember, I emphasized there were three subunits on PDH. One is E1. The next one is E2. And the last one is E3. The reason I emphasize that is because the conversion of pyruvate to acetyl-CoA is a three-step reaction. The first step is going to require pyruvate binding to the enzyme's active site, but it doesn't bind to the enzyme. It binds to thiamine pyrophosphate, vitamin B1. So B1 needs to be in that active site to initiate pyruvate binding to the enzyme. After that, what's left is an acetyl group, and that acetyl group now, which is in this box, is handed off to E2, and then E2 does something to E3, and then what we magically get out at the end is acetyl-CoA. So this is the level of understanding that I'd like you to look at. You've got E1 requires thiamine. If you have a thiamine deficiency, what's going to happen to E1 activity in PDH?
Non-existent.
It's not going to be able to bind the acetyl group of pyruvate. And if it can't do that, the concentration of pyruvate increases. Because thymine inhibition is the same thing as having a genetic mutation on this enzyme or lack of the enzyme. And if pyruvate builds up, what happens there? Where does it go? Yeah, it goes to lactate and lactic acidosis. So add on to the weakness, pain, muscle cramps, tired, blah, blah, blah. Add on there that a thiamine deficiency can lead to lactic acidosis because you've got a block in pyruvate processing. Now, to address these clinical symptoms, how does fatigue and tiredness result from a thiamine deficiency? Are you not producing energy at all? Sure, some, right? Up to pyruvate. This is glycolysis. So you're producing a couple of ATPs. Isn't that enough? So what's going to happen to skeletal muscle when it continues to generate these two ATPs and it has a thiamine deficiency? It's going to push that pyruvate even more into lactic acidosis, into lactate production. That will cause the muscle cramps and muscle pain. Because we're blocked at this step with a thiamine deficiency, that means the concentration of our ability to make ATP is depressed. Okay, so that's B1. Now B2. I'm just going through B1 through B7 today. We'll do B9 and 12 tomorrow because that's kind of a special case. So B2 is riboflavin. Deficiencies are rare. Seldom ever have a deficiency in riboflavin. But if there is a deficiency in riboflavin, this is where the problem is going to precipitate. FAD production, because this is flavin adenine dinucleotide. This is your flavin molecule right here. And so this is the backbone forming the flavin adenine dinucleotide FAD and flavin mononucleotide FMN. So where do these cofactors show up? Where does FAD, what's a very important enzyme that has FAD? Yeah, succinate dehydrogenase. What about pyruvate dehydrogenase? FAD is part of the bound of the E3 complex. And so the FAD is going to be critical in the activity of pyruvate dehydrogenase in order to regenerate the enzyme to keep that reaction going. So those are two sites that the FAD is important. So what about FMN? Have you seen FMN anywhere?
Complex 1 in the electron transport chain.
Yeah, the electron transport chain. And it's an electron carrier. So anytime you get something that can be reduced or oxidized, it's going to be part of an energy generating pathway. And so if you have a deficiency in vitamin B2 riboflavin, what do you think the symptoms could be? Same thing. we're in an energy producing pathway. So this is going to create a decrease in energy and all those horrible manifestations. Fatigue, tiredness, weakness. Okay. Now vitamin B3. B3 is niacin, nicotinic acid. The disease associated with this is pellagra. Now again, connecting pellagra with niacin and B3 is critical for the step 1. I'm sure you've seen that on first aid. But if I have a question about a niacin deficiency, I'll mention it's part of that disease, pellagra. Skin rashes, peeling, flaking, lots of skin problems. So deficiency in niacin is going to be a deficiency in nicotinamide generation. So NAD levels, NADP, and here's the NAD molecule. This is from niacin. The rest of the molecules constructed on there to give you your NAD structure. When we talked about NAD is energy, NADP is used for biosynthesis. The only difference between these two is if there was a phosphate right there, this would equal NADP. That's the only difference between the molecules. But they both come from the same thing, from niacin. Now, some of the clinical symptoms are skin rashes, peeling, and flaking. What if I told you that these individuals with a B3 deficiency also had diarrhea? So how does a deficiency in the nicotinamide molecules lead to skin rashes and diarrhea? What's that connection?
Turning over cells.
Yeah, turning over cells. So rapidly dividing cells, skin cells, intestinal cells, do they require energy? You bet. Do they require biosynthetic power? You bet. And so a deficiency in B3 is going to compromise their energy and biosynthesis potential, leading to interruptions in cellular replication. Now I put this here because I had no idea myself. We're going to go from B3 to B5. And for some reason, I never asked myself until this past summer, what happened to B4? Well, B4 was a designation given to several chemical compounds. This was in the old days when vitamins were being named, and B4 was considered often one of these 3 different compounds. And that's no longer, these are no longer considered a vitamin. They're cofactors, but they're not considered vitamins. So that's why there's no B4. Okay, we're good on B3? So B5. Again, this is another one, like B2. Deficiencies are rare. So nothing you probably ever have to think about or worry about. But vitamin B5 is pantothenate. And pantothenate is necessary for coenzyme A synthesis. This middle portion of the coenzyme A molecule is pantothenic acid. The rest of the molecule is the adenosine nucleotide structure. And then this very end right here, this reactive thiol right there, that's where we generate our acylations. So when we draw acetyl-CoA, the acetyl portion is very, very tiny compared to the CoA. And in that CoA, we require pantothenate in order to construct the coenzyme A. So even though it's a rare deficiency, what would the symptoms be? Death. Tiredness, fatigue, probably death. We need acetyl-CoA for everything. We need it for glucose metabolism, lipid metabolism, protein metabolism. So this would be a very severe deficiency. But fortunately, it's rare. Okay, that's a boring one. The next one is really fun. Vitamin B6, pyridoxine. Now, these deficiencies are rare. However, unlike B5, pantothenate, which doesn't have a lot of questions associated with that thing, this one can have a lot associated. The pyridoxine cofactor, B6, is critical for several different reactions. The most important one for this block is involved in the aminotransferase activities. Now, if you have a deficiency in aminotransferase, would you expect to see weakness, sleeplessness, peripheral neuropathy, and personality changes? And what would be causing that? Yeah, hyperammonemia. Now, here's what B6 does. So we have to revisit our transamination reaction. So I talked about transamination before of saying when alanine comes in from skeletal muscle into liver, the first thing liver is going to do is a transamination reaction to convert the alanine to pyruvate, and then it's going to have a place for that nitrogen. It's going to put this, of course, in the liver is going to go on to gluconeogenesis. Now, we're moving in this direction from alanine to its respective alpha keto acid, which is pyruvate, which means that now this nitrogen on alanine is a donor and it has to go to a recipient. The recipient on this is going to be the transaminase with the pyridoxal phosphate. All that is a phosphate on pyridoxine in the active site. And what we're going to do is we are going to transfer this nitrogen to this particular carbon on the pyridoxine. When we do that, here's the nitrogen. So the nitrogen gets physically transferred from alanine to the pyridoxal structure in the enzyme. But now, because enzymes need to be regenerated to continue their use, we need to convert the pyridoxamine, the nitrogen-bound pyridoxine, back to the unbound form. In order to continue this reaction and continue alanine processing. So what we're going to do now is we are going to take this nitrogen and we're going to put it onto this alpha keto acid, which is oxaloacetate. And this is all occurring in the cytoplasm. And here's our nitrogen to form which amino acid? Aspartate. And then the aspartate is going to be added to citrulline. And then this will form argininosuccinate. And then on to forming urea. What's the first donor of urea? The ammonium ion. And how is that captured? CPS1. In the cytoplasm? In the mitochondria, yeah. So that's the first direct donor of urea, and then aspartate is the second donor to make that second nitrogen on urea. Okay, so this is where B6 deficiency comes in. So now if you have a B6 deficiency, what would be the clinical symptoms? You're going to have hyperammonemia leading to personality changes. We saw that in that young mom in our case. Peripheral neuropathy, because we're decreasing glutamate synthesis. Sleeplessness. If I had high ammonia levels, I don't think I'd want to sleep. How do you get weakness with a B6 deficiency? If B6 deficiency means we turn down the activity, shut down the activity of our transaminase, so what can we not do? Break down amino acids, yeah. So now what we've done is compromise a very powerful energy source for ourselves. That'll lead to weakness. Will it lead to hypo or hyperglycemia? It can lead to hypoglycemia, right? Because now all of that alanine that's coming into the liver cell, that pyruvate that is going to be used as a substrate for glucose synthesis, now the alanine gets stuck because we have a B6 deficiency. Okay, questions on that? Does that help with the transaminase stuff a little bit more? Kind of putting in a little bit of the mechanism? Okay, so now there's a couple other places that B6 is required. These aren't necessary for me, but I think Dr. Burge will talk about these. First one is catecholamine synthesis. We're going to take tyrosine, and we're going to make three important products from tyrosine. One is dopamine, the other is norepinephrine, and the third one is epinephrine. So that's all going to come from tyrosine metabolism, the adrenal medulla. And this particular enzyme right here is B6 dependent. So this pathway, this enzyme, not important for me, but just to kind of give you a heads up for Dr. Burge when he talks about catecholamines, that we're going to have another B6 dependent enzyme in here. The other one, same thing. Not important for this portion of the block, but synthesis of serotonin and melatonin comes from tryptophan. And this particular enzyme, this decarboxylase, is also vitamin B6 dependent. So there's the transaminases, those are the big ones. And then the other one is these decarboxylase enzymes. Oh yeah, and there's one more.
Are you saying you don't want us to memorize
No.
the pathway? We just need to know that enzyme, this pathway, that vitamin.
Yeah, just that the decarboxylase enzymes in tyrosine and tryptophan metabolism that leads to the catecholamines and serotonin and melatonin, those decarboxylases are B6 dependent. Again, it's just a recognition of an enzyme with a vitamin cofactor. But there won't be any questions on this exam on Friday that has to do with catecholamines or serotonin and melatonin. I'm going to leave all of that for Dr. Burge if he feels that's important. So here's one more. Glycogen phosphorylase. This is also B6 dependent. So here's our pyridoxine. This would be in the cofactor in the glycogen phosphorylase enzyme. And it's going to mediate this reaction. So this time here, this is the other end of pyridoxine. The phosphate group on the pyridoxine, making it pyridoxal phosphate, is going to cause a little bit of a change in inorganic phosphate such that it now attacks that bond between glucose and glycogen, releasing glucose hydrolysis or phosphorylysis leads to 1 phosphate. And then, of course, this goes on to glucose 6-phosphate. But that phosphorylasis reaction is adding inorganic phosphate to cleave that glucose off in glycogen phosphorylase. And that requires that pyridoxal phosphate. Now again, the reaction mechanism and all that is not important. This is just so that you can kind of lay your eyes on it for one time and see how that works. And hopefully that will help associate pyridoxal phosphate with glycogen phosphorylase activity. Now our last one is B7, biotin. So there's a lot of symptoms with biotin deficiency. Lots of non-specific ones. Fatigue, loss of appetite, muscle pain, nausea, weakness. Hair loss is a big one. I make sure I take plenty of vitamin B7, just, you know, but mine is genetic. Did I mention my grandfather was bald as a billiard cube at 25?
Oh,
Yeah, I mentioned that, yeah. Can you tell it's a sore spot on? No, anyway. So the biotin deficiency is quite diverse. So what does biotin do? Well, biotin is necessary for carboxylation reactions. These are the two major enzymes, acetyl-CoA carboxylase. That's the enzyme that converts acetyl-CoA to malonyl-CoA for fatty acid synthesis. The other one is pyruvate carboxylase. Now, biotin has a role in quite a few other enzymes, but these are the two major ones that have major clinical effects. So here's what biotin does. This is the biotin molecule, and this is what it does. It will bind CO2. Of course, the CO2 is coming out of the aqueous solution, this bicarb, and it will bind to the biotin molecule, and then this can be donated in reactions. So a good example of this are these two. Pyruvate is a carboxylation reaction that requires the addition of CO2 onto pyruvate to give you oxaloacetate. Same thing with acetyl-CoA. This is another carboxylation. So we simply have to have a donor of the CO2 in order to facilitate this. And that's what biotin does. Now there are other decarboxylation reactions that can also require the CO2. So in this case, the CO2 is being donated to biotin. So biotin is involved in what's called one-carbon transfers. And one carbon almost always means CO2. So whether we're going to add a carbon or extract a carbon. We'll see tomorrow about vitamins B9 and B12 that are going to have similar types of carbon transfer abilities. But this one here is involved in CO2 transfer. Yes?
So does the CO2 from bicarbs cause the donation of CO2?
Yeah, the reaction here is to take the CO2 out of the circulation, and it's converted into CO2 and it binds to the biotin molecule itself. So it's not to think that CO2 is a gas that's coming out of the circulation. It's CO2 is always coming out in these reactions as bicarb.
So that CO2 is that CO2 group on the biotin?
Yes,
Oh,
exactly.
okay.
Yes. And this, of course, is coming from respiration,
Oh, okay.
from TCA cycle.
Okay.
Now, another important point about biotin is raw egg whites. Since we're in Nutrition Week, there are some people that eat raw egg whites. Why would you eat raw egg whites? Protein, because you're crazy.
Oh, it's protein. Or rocky.
Have you ever seen people that want to really take a lot of protein intake, but they don't want to go to the health store and get the proper nutrition and diet? They will take a whole bunch of eggs, put them into a blender, like a dozen, mix them all up, and bottoms up. To me, it's like eating oysters. Yuck. But it's a huge source of protein. Now, the problem with that is that egg whites, the main protein in egg white is ovalbumin. So in our blood, we have albumin. Chickens make albumin, but it's called ovalbumin. That's the majority of the protein. The problem is that we also have another protein called avidin. Avidin binds to biotin with almost the affinity of a covalent bond. And so once biotin is bound to avidin, that biotin is no longer biologically available. There's no way to dissociate those two because of the strength of that bond. And so how do you solve the problem? You cook your eggs. Pretty simple. The heating of eggs will cause the denaturation of avidin. And if avidin is denatured, it can no longer bind the biotin. Yes?
What is ovalbumin doing to avidin?
Ovalbumin is the major protein in egg white, and avidin is just it's protein in there, but a smaller quantity.
Okay.
Yeah. So ovalbumin and avidin don't have any interactions. So the heating denatures the avidin, and it actually reduces the amount of binding and tying up of the biotin. Okay, so biotin we can get from many different sources. One of them actually is egg yolks. So what's the problem here? Well, if you eat a whole egg and you don't cook the egg white, the avidin will bind the biotin and you cannot absorb the biotin, leading to a biotin deficiency. So by cooking our eggs, we can now have the avidin denatured, and the biotin now is free, and we can actually absorb it and internalize it and have access to it. So that's where biotin deficiency comes up from raw egg whites.